Okay, so this is chapter 6, which is entitled The Molecules of Macrophages. And of course, me being a T-cell person, I've sort of tried to avoid macrophages my entire life. But I wasn't able to do that because they were found to be essential for the generation of an immune response. And I think I've said a little bit about that before, but so now we're going to go into that in detail. And there's a lot of details. Macrophages are very sort of ubiquitous cells. They're in every tissue in the body. They float around in the blood as pro-macrophages that are called monocytes. And the word macrophage means big eater. And so what they do is they phagocytose particles. And that's the major mechanism whereby the immune system rids the body of foreign materials. So the macrophages are essential for that function, for phagocytosis. And that was originally discovered back in 1881 by Eli Mechnikov, who was a Russian scientist who had to have been in the nobility anyway, because he vacationed in Napoli in summertime, and or maybe the wintertime, I can't remember which. And he was studying, he was a biologist, and he studied starfish larvae, and he was looking at them under the microscope. You know, the microscope had been around since the 1600s, but it really didn't come into use until the 19th century, really, at least with biology. And he noticed that in the starfish larvae, that if he put foreign materials on his slide under the microscope, that there were cells there that would just gobble up that stuff and do away with it, essentially. And so that was the beginning of phagocytosis. And it was really the beginning of what became known as cellular immunology. And that was the going feeling about the immune system and immunology for several decades. And actually, Louis Pasteur, when they started the Pasteur Institute in the 1880s, he recruited Mechnikov to come to the Pasteur Institute to be the chief of service there. And so he arrived in 1885, just when the institute was opening. And so it was a big deal. It wasn't until 1890 when the rival guys in Berlin that I talked about earlier, Robert Koch, and then his lab and institute in Berlin, they discovered the phenomenon of antibody activity in sera, in the humoral, and that was the beginning of humoral immunity. And so the humoralists and the cellularists then warred for about the next century, as humans are wont to do. The macrophages, the study of macrophages it wasn't until the advent of the whole 2 decades, the 20 years of cellular immunology, when that took off between 1960 and 1980. And as I said in the last episode, in 1963, Niels Jerne developed the plaque assay for the generation of antibody-forming cells by mixing mouse spleen cells with sheep red blood cells, and then putting them in soft agar and showing that there were little lytic plaques around each B cell that was making the antibody that would lyse kill the red blood cells. It was soon thereafter, in I think 1967, when Don Mosier from the Scripps Institute in California showed that if you took the macrophages out, then you didn't get any antibodies. So that was basically the first experimental inkling that these things were sort of important for the immune response. And then in 1968, a friend of mine, Joost Oppenheim from the NIH, showed the same thing with human lymphocytes and macrophages, monocytes, that if he purified out the lymphocytes and only put them into culture with mitogenic lectins like PHA, phytohemagglutinin, that the proliferative response wouldn't go away, but it would be markedly diminished, and 90 to 95%. And so there seemed to be something that was important. And that basically, by the end of the 60s, everybody knew that. And then in 1970, there was a fellow by the name of Fritz Bach from the University of Minnesota, and he showed that for the proliferative response, that he could culture macrophages separately from the lymphocytes for a while, and then take the supernatant, the culture media, from those cultured macrophages, and he could replace the macrophage effect. And so that was in 1970, and then in 71, there was another group from UC San Diego, Dick Dutton and Mike Hoffman, who showed that you could do the same thing with the urinary plaque assay, the Michel Dutton assay, whereby you could replace the macrophage component with macrophage supernatant, soluble stuff. And then in 1972, so things were moving along pretty fast in the early 70s, there was a fellow by the name of Yigal Gary, who came from Israel to work at the National Institutes of Health in Bethesda. And he did a series of experiments. Actually, he wasn't in Bethesda yet. He subsequently got there. Before that, he went to Yale, and he worked with a fellow by the name of Byron Waxman. And he showed, there were two papers that came out back to back in the Journal of Experimental Medicine, whereby he really nailed it. He showed that there was this factor activity that would come out of macrophage cultures, and if you used stimulants such as lipopolysaccharide, LPS, which is the major component of a lot of bacterial cell walls, that you could get a huge amount of this soluble activity. And he coined a name for it, and he called it lymphocyte activating factor. And that was then abbreviated LAF. We always have to have our acronyms, and that's when I think I said before in one of my videos that in our lab we didn't want to deal with these macrophage things, and so we called LAF a joke. That's what it was to us. But Yigal Gary's two papers were very important because they not only show that the macrophage made this factor, they made it in response to these stimuli, and he also devised an assay for the factor, and he showed what the factor was doing was activating T cells, and in the assay he devised used thymocytes as the target cell, and thymocytes being almost 100% T cells. It was just a lazy man's way to get a macrophage depleted culture of T cells, and then you could add things to the T cells and do your experiments that way. And this is sort of the reductionist science approach to life. And I was at the NIH at that time, the NCI, the National Cancer Institute, and I was working with my mentor was a fellow by the name of Michael Martinet, who had come from the Scripps Clinic, and he was interested in lymphocyte proliferation in response to antigens and mitogens and that sort of thing. And so I started purifying T cells separate from macrophages and then adding them back again. And I had my benchmate, and actually the guy that recruited me to come to the lab was a fellow by the name of Lenny Chess. And he was a New Yorker from Columbia Medical School. And he and I were peers as new recruits into the NIH, and of course I think I described before it was during the Vietnam War, so we were happy to be in Baltimore rather than Da Nang, and so I purified out the macrophages and then showed that when you activated these cultures, and Lenny was working with two different substances, one called poly-AU and another one called poly-AC, which were synthetic RNA molecules. And these molecules were interesting to us and to Lenny because of the fact that they were like LPS. They had been shown to be adjuvants in vivo. In other words, if you immunized a guinea pig with an antigen and then at the same time delivered either LPS or poly-AU or poly-IC, that you get a markedly increased antibody response. And what we showed was that it also enhanced the proliferative response of T cells in vitro. So that was 1972. Then it wasn't until 1974, only a couple of years later, that another fellow got involved in the whole thing, and his name was Charles Dinarello. And he had been also down at the NIH, and he had been influenced by a whole group of investigators at Yale who were the first people to describe soluble products that they thought came from macrophages and monocytes that caused fever. And so he was involved in a laboratory at the NIH that was interested in what was causing fever. And so you can imagine fever being a very important clinical symptom or sign. And yet we didn't have a clue how the whole process worked, how the fever was generated and why the temperature would go up and then it would come back down again. And when it came back down again, that was a good sign. Either that you were going to get better or you're going to die. Either one of those two things. So Charles learned how to do the rabbit fever assay that had been developed at Yale. And that entailed taking substances that you thought could cause fever, and the experimental word for that was it was pyrogenic, or the substance was a pyrogen. And because the idea was at the time that it came from inside the body, it didn't come from say a bacterial infection or a viral infection. It wasn't the bugs that were causing this fever. It was an endogenous pyrogen. And so the hunt was on in Dinarello's group to find what was causing this fever business. And he had an assay for that where you would take your substances, inject them into rabbits, and then you could measure their temperature, their body temperature over time thereafter. So the rabbit fever, of course, rabbits are pretty big animals. Most immunologists are used to working with little mice. The rabbits are great big things, so they're not as easy to deal with. Plus, they bite. So he started out in 1974, and he was trying to purify this endogenous pyrogen. The abbreviated name is EP. And he did. He was successful. He purified a lot of it, and he injected it into rabbits, and he made anti-sera activity that he could show was due to the immunoglobulin molecules and so forth. And then he used that serum to purify the endogenous pyrogen. And he put it into a LAF assay on top of the thymocytes, and lo and behold, EP scored positive in the LAF assay. So then the question was, so that was 1977, and the question was, so, is EP the same thing as LAF? And right at the same time, as you may recall, in 1977, we published our first paper on the long-term culture of cytotoxic T lymphocytes, or the CTLL. And as you may recall, it came out in Nature on Bastille Day in 1977, which I always really liked. And so we had also that we started to publish papers on T-cell growth factor activity. And one of the reviewers in our very first paper, he said to us, "How do you know this isn't LAF?" And I said, "Well, what's LAF? I never heard of it before." And so now we had to figure out whether or not whether we just rediscovered the wheel, and something that these other people had already known about, and so forth, which wouldn't have been so good for us. And so the thing that we had devised was an assay for T-cell growth factor. The target cell were not thymocytes, as in the LAF assay. The target cells in our assay were these long-term and cloned T cells and functional T cells. And we knew that whatever we called T-cell growth factor, supernatant at the time, we knew how to grow our T cells in that stuff. And then, so we started to collaborate with Joe Oppenheim and another investigator down at MD Anderson, Larry Lockman, who were purifying LAF, and they sent us some of their purified samples. We put it in the TCGF assay, and it was absolutely negative. Cells died. And that's when we started saying that LAF was a joke. On the other hand, if we took our semi-purified, we were trying to purify TCGF. If we put that into the LAF assay, it was positive. So then so what was the story here? And To make a long story very short, we did a series of experiments in collaboration with Oppenheim and Lockman, and showed basically that what was going on is that you could stimulate macrophages with LPS and poly-IC or poly-IU, take that supernatant, and then put that onto T cells, and those T cells then would make a lot more T cell growth factor activity. So, and that was then became the rationale to call LAF interleukin 1 and TCGF interleukin 2, because it seemed that LAF was working in the sequence before TCGF or IL-2. And that brings us to 1979 and 1980. So over the course of a decade, between 1970 and 1980, we finally had a pretty good handle on this macrophage mitogenic activity and why it was mitogenic for T cells. It was mitogenic because it caused the T cells to make the mitogenic activity. So then the race was on at that stage of the game to find out, to find the molecule responsible for the whole thing, for the LAF activity. At the same time, we were focused on TCGF, the TCGF molecule, and purifying that and so forth. And I've already recounted that story. So this time is the LAF endogenous pyrogen story, and the principal actors in this whole thing were Joe Oppenheim on the one hand, and one of his postdocs by the name of Stephen Meisel. And Joe and his students had identified a macrophage cell line that would seem to make about 10 times as much LAF activity as the normal macrophages. And so Steve Meisel took that macrophage cell line, because you could get a lot of those cells, you just grow them up in culture and so forth, stimulate them with LPS, lipopolysaccharide, and then take the supernatant, and then he took that and tried to purify that biochemically with biochemical separative procedures that basically at the time separated things on the basis of molecular size or molecular charge. And those were the two different things, and so that you could put things through a long column and take fractions off the column and find out where it was coming out, they would separate as to the big molecules would come out first, and then trailing behind with the smaller and smaller and smaller molecules. Then you could take those, semi-purified peak of that activity, and then you could put it onto other biochemical procedures that now would separate the molecules according to charge. And with those two things, you could pretty well sort out things. The problem was, most of the time you were starting with what seemed to us at the time huge amounts of conditioned medium, like a liter. So a liter is like a quart. So that seemed like a lot to us, but that was minuscule. We needed 100 liters, And so to really end up with anything because each time you put it through these processes, you lose stuff. You can purify things down to purity, but you don't have anything left to deal with. You can't then take it to the next steps, which are to show that, our pure molecule does this, or our pure molecule does that, or these are the characteristics of the pure molecule. What Steve Meisel did is that he did the same thing that Dinarello had done, but he used a goat, and he purified his LAF as much as he could, and he stripped it into some goats, and lo and behold, he got a good antibody, anti-sera out of that, and on the basis of that, he went to the next step. Meanwhile, Dinarello was using his antibody to purify human. Meisel was working in the mouse, and Dinarello was working in the human, and so Meisel collaborated with some people at Hoffman LaRoche and a fellow by the name of Peter Lemedico, and they devised a system so that they could, with their purified LAF activity from the mouse, they could deduce the amino acid sequence, and from there they could work backwards to clone the gene. And they cloned a gene for mouse LAF, and almost simultaneously, Dinarello, working with a group at MIT, they cloned a gene for human LAF. Meanwhile, there was another group out in Seattle that was working with us to do the same thing. And over the course of 1983 and 1984, in 1985, it was 2 or 3 years, the genes were cloned, and it was discovered that there was not just one molecule that had LAF activity, but there was at least two. And so they were named interleukin 1 alpha and interleukin 1 beta. And it turns out beta was present in like 10 to 20-fold higher quantities in any of these supernatants than the alpha was. And they were not very homologous by their sequences. They were only like 25% homologous, but they were homologous enough, and the overall structures of the molecules were very similar. So that by the mid-'80s, that whole situation had resolved. And not only that resolved in the fact of these mitogenic factors coming out of macrophages were now real molecules, but in addition, they had solved the problem that between all the T-cell factors and the mouse and the macrophage factors. And of course, in the last video, I went through a lot of the experiments that had been done trying to find out how the generation of antibody-forming cells were influenced by these soluble factors. And we talked about interleukin 4 and interleukin 5 and interleukin 6. And also the fact that interleukin 3 was a misnomer. It was not in the immunological family, it was in the hematopoietic blood-forming cell family of molecules. So the immunologists just forgot about interleukin 3. You know, it wasn't one of our guys. So that was it. By the mid-'80s, we had all these interleukins up until number 6. But there was another thing about macrophages, and that was very curious, and that was, it seemed as if there was a cellular component of macrophages that was necessary. You couldn't replace them, the presence of macrophages completely, and still get a T-cell proliferative response or still get an antibody-forming response. You had to have, there's something that there was touchy-feely going on between the macrophages and the T cells. In the 60s, so we have to go back from mid-'80s to the mid-'60s, essentially. There was a fellow by the name of Baruch Ben-Asaraf. I may have mentioned him before. He was originally a Moroccan fellow his family emigrated during the war to Venezuela, where he grew up, and he did his university and medical. He went to medical school in Paris, and he did his postdoc there, and then he came to New York and set up his lab in the 60s. And he started looking at not the antibody question had just been solved. Now we knew what antibody molecules were. They were these things called immunoglobulins. We knew the structure and so forth. And it was before the cytokines and the lymphokines and the lymphodrek came into being. And so he thought he'd focus on the antigens. The question is, what's an antigen? And how do you become an antigen? And so he said to himself, well, antigens are anything in nature that are foreign and that can stimulate an antibody response. And that's what the definition of an antigen is if you look in the dictionary. So we have antigens that stimulate antibodies, and antibodies bind to antigens. Ben-Asaraf thought, well, let's just not take, up until that time, everybody had just used things that were around, like bovine serum albumin that they could get from the slaughterhouse from cows, or they could get pure molecules. But Ben-Asaraf said, well, why don't we see if we can make synthetic peptides in the laboratory. By this time, you could synthesize proteins by adding amino acids in a long chain form, one to another and one to another. And he showed in 1963 that he could take a synthetic peptide that he made in the laboratory, and he could cause an immune reaction in guinea pigs. He could cause what they called a delayed type hypersensitivity reaction, so that when you put the thing under the skin and you waited a day or two, the skin then would get red hot, swollen, and sore. You'd have an inflammatory reaction. And then just a few years later, in 66, one of the investigators by the name of Stuart Schlossman showed that you only need you needed at least 6 to 8 amino acids in a row to cause this. If you had less than 6, you wouldn't get that reaction. Nobody really understood why that was, but those were the data. Then in 68, in 66 also, Benacerraf had shown he worked in guinea pigs all the time. And he had 2 different strains of guinea pigs that were histo-incompatible. They were not totally inbred like the mice were, but they were enough different. There was strain 2 and strain 13, they call them. And I worked with those guinea pigs for a while. And he showed that if you gave them different synthetic peptides to the strain 2 and compared it to the antibody response or the DTH response to the strain 13, that there were high responders to some peptides and low responders to some peptides. And it was always that way. So somehow the genetic background of the animal had something to do with how well the immune response worked. Well, that's what Benacerraf ultimately went on to win the Nobel Prize for in 1980, just to give you sort of a thing on that. But one of the most important things that happened soon after Benacerraf reported that in 1966 was in 68, Hugh McDivitt at Stanford was working in mice. The guinea pigs were okay, but they didn't have tails, so you couldn't pick them up very easily and so forth. And mice were much easier to work with, and they were the favorite animal of the immunologist. And so he took the whole idea of these synthetic peptides, and he could find and there were a lot of different mouse strains that had already been inbred since the 1930s. So this was 68, so that's like 40 years later. And so we had all these different mouse strains, and they knew what the genetic differences of all the mouse strains were. So he immunized these different mouse strains with the same synthetic peptide, and he found that just like Benacerraf had shown in the guinea pigs, he showed in the mice that there were low responders or absent responders and responders. The most important thing he showed was that mapped genetically, because he knew the genetics of his inbred mice, to the genes that controlled the histocompatibility between one strain and another. So if you had two different strains that were histo-incompatible, they would reject each other's skin grafts or kidney grafts or any graft that you wanted to do. And that was the test then, that they were histo-incompatible. If they were histocompatible, they'd be like identical twins at the genes over the loci in the chromosomes and in the genes that controlled this genetic trait. So then that was very interesting, but it was also, an enigma. I mean, so what's that all about? And actually, that was just about the time that I entered into the business, and I never understood what these guys were so excited about. I said, whatever. Why is that so important? Well, and so in the 70s, things got sorted out a little bit better so that was 68, and between 68 and 1973, over a period of 5 years, more and more experiments were being reported, but they were basically the same thing. Then in 1973, there was a group at the NIH that showed that in vitro, to make a T-cell proliferative response, that the macrophages and the T-cells had to be from strains that were histocompatible. And if you use 2 strains that were histo-incompatible, then you wouldn't get a proliferative response. And then almost at the same time, Benacerraf showed, with one of his postdocs by the name of David Katz, that in order to get an antibody-forming response, the T cells and the B cells had to be histocompatible. So that was called major histocompatibility complex restriction, or MHC restriction of the immune response. And the question is, well, why is that? Then people thought about that for a while, but it wasn't until a few years later, in 1974, actually only 1 year later, there was a group down in Australia. There were actually, one of them was an expat, and his name was Rolf Zinkernagel, and he got together with another postdoc by the name of Peter Doherty, who was an Australian. He was one of those descendants of the Irish when they sent the Irish down the penal colonies. Zinkernagel got together with Doherty, and they were interested not in antibody-forming cells or T-cell proliferation. They were interested in cytotoxicity that we talked about in one of the other videos. And they showed through a series of experiments in 1974 that the T cells and the target cells that were going to be killed by the T cells had to be histocompatible. And so that was important because of the fact that meant that you didn't have to worry about macrophages necessarily, and you didn't have to worry about B cells either. That the T cells and it was a T cell and probably a T cell antigen thing. That led then to so the conclusion of all three of these experiments, one done with T cell proliferation, one done with antibody formation, and the other one done with cytotoxicity, meant that this must have something to do with the T cell receptor that I've already talked about in terms of solving the molecular problem. But this led to the whole excitement over the T cell receptor, because we were getting closer and closer and closer to what this thing might be. And so they came up with two hypotheses, the one receptor hypothesis or the two receptor hypothesis. The one receptor hypothesis said that whatever the antigen, this peptide was that Benacerraf and McDivitt and these guys had talked about, this antigenic peptide, you had to have a receptor for the antigenic peptide, but that somehow had to be involved with a receptor for the histocompatible molecule on the surface of the different cells that they were mixing together. So there was the one receptor hypothesis that meant the antigenic peptide and the MHC gene product were the same, or pretty closely, together. Or there was a two receptor hypothesis that said that there were two different receptors that had to be recognized, but they were two independent receptors. That then led to, at the end of the 80s or 70s, in 1978. So everybody, they were fighting. All the immunologists were fighting about what was the one receptor. Somebody did an experiment, said it was one receptor, another that it was a two receptor thing, and so forth. And so Benacerraf said, he published an opinion article in the Journal of Immunology in 1978 that really, I think, got him the Nobel Prize that they got only 2 years later in 1980. And that was, what he said is that what's going on here between the T cells and the macrophages, or between the T cells and the B cells, or between the T cells and the target cells, is the peptide. He said, I'm not going to tell you whether it's the one receptor hypothesis or the two receptor hypothesis, but the antigenic peptide has to be co-recognized with the recognition of MHC, of the MHC encoded molecule. That sort of, the reason it was important is that it cleared the air a little bit. It didn't really solve the problem, but it cleared the air. And so, just to go back in terms of the descriptions that I gave earlier about Ellis Reinhardt's coming in 1982 and 83, so it was just a few years later after the end of the 70s, he really sort of did a slam bam, thank you ma'am, this is it, and now he walked away and the dust was cleared, so that was that. And so the question then is that as time went on in the decade of the 80s, the question was, what is this MHC gene product or products? What's the molecular basis of that? That turned out to be a huge story and a huge genetic story. And that was another one of the things that was really, it essentially would preoccupied immunologists for the entire 70s and most of the 80s. And the investigators that got involved in that were interested in trying to get at the molecules. And there was one fellow by the name of Jack Strobinger at Harvard, who did a lot of the yeoman's work. He was a biochemist and an immunologist, and they basically, over the course of several years, grew up cells that they knew had MHC gene product on the cell surface. Liters and liters of them, purified the cell membrane molecules, identified the MHC molecules, and then got together with some gene cloning jockeys, and they started to take the information they had from the proteins and to get to complementary DNA molecules that would do that. And they found some of the first molecules. They found evidence that then there was other groups involved too. Leroy Hood from Caltech, and I think it was Francois Korilski from Paris. They all did very similar experiments and found very similar things. So what they found was that there were molecules that seemed to be there seemed to be a lot of them in the genome. And there wasn't just one gene and one gene product. There seemed to be all kinds of genes and all kinds of gene products. And they got classified they were classified at the time already into class 1 versus class 2. And they found around 30 to 40 genes that were in the class 1 category, and they ultimately found a similar kinds of genes, but similar numbers of those that were in the class 2 category. And then Strobinger got together with people who were crystallizing proteins, and when they could get enough using the genes to get the gene product, when they could get enough of the gene products, they crystallized first the MHC class 1 molecule complexed together with the T-cell receptor, and this was by 1987. So with just a few years after the T-cell receptor had been elucidated, essentially, they got a picture. The thing about structural biology is that if you're successful in getting a huge amount of whatever your protein molecule is, and if you can get it into a concentrated solution, and then if you can get adjust the characteristics of the solution so that the proteins salt out. And it's just like when you super saturate a solution with sugar or salt, and you get crystals that go down to the bottom. That's what you can do with proteins. And then once you get those crystals, you can take those crystals and shine X-rays at them. And when the X-rays hit the crystals that are all arrayed in a very uniform molecular pattern, they will deflect the X-rays so that then you have an X-ray film behind it, so that they sort of spray off. And on the basis of that, you can, with mathematical formulas and so forth, deduce which atoms are causing the deflection. And on the basis of that, you can put together a picture of the molecule. So for the first time in the history of immunology, and this was an aha moment, if there ever was one. It was published on the front page in Nature and so forth, of the MHC molecule and the T-cell receptor molecule binding together. And the interesting thing was that the MHC molecule it looked like a hot dog bun. So it had two alpha helices, helical twists of the protein, and in the middle was where the antigenic peptide sat. And then the T-cell receptor would come down and abut that whole thing, and that then was, I think, put together, like 40 years of wonderment about what's going on here, because finally they knew that this is what was happening. And of course, once the T-cell receptor then was activated by the MHC peptide antigenic situation, then the T cell would become activated and it would make cytokines like interleukin-2 and so forth and so on. And that's how you would generate an immune response. Also in the 80s, simultaneously with all those experiments that were going on trying to purify MHC gene products and understand that whole thing, the other question that was generated by the experiments done in the 60s and 70s was, well, how does the macrophage make these peptides? And how does it put them, how do they get together with the MHC molecule? What's going on in that whole thing? And there was another fellow that was a major player in this business, and his name was Emil Unanue, and he was originally from Cuba. And I've been to Cuba at least 10 or 11 times, and so I sort of liked him. He was a very flamboyant Latino kind of a guy. But the thing about him is that he was a young guy, and he essentially came from Cuba and got out of Cuba and became a postdoc or a young investigator in Baruch Ben-Esraf's lab up at Harvard. And rather than get involved in the T cell and B cell end of things, he started in on very early of trying to understand and figure out how the macrophage was able to phagocytose these molecules on the one hand, or all the microbes. And then what happened? And basically what they found over the time is that the macrophage had all these granules in it that were digestive granules, and these things had all these digestive enzymes in them. They would break up the whatever the foreign particles were into smaller and smaller units and so forth. Ultimately get down to very small peptides and so forth. And he did a series of experiments in the 80s that essentially nailed it down to what was going on is that the lysosomes, which are some of the granules in macrophages, would break down the antigenic microbes into molecules and then into submolecules. And there was all kinds of cellular trafficking going on inside the cell, so that the peptides that were resulting from this digestive process would find their way to either MHC class 1 molecules on the one hand or MHC class 2 molecules on the other hand. And they then would traffic to the membrane and be placed on the membrane, with the hot dog bun and the peptide in there, and that was it. And so essentially by 1990, we, meaning the community of immunologists, had solved a lot of the issues that had grown out of the cellular immunology years of the 60s and the 70s. I'll stop there.